Great, I will go ahead. So good afternoon, everyone, and you're all very welcome to our Epilepsy in English event. And we're very excited and honored to close the Epilepsy Island Conference. So my name's Gareth, and I'm one of the co-founders of Epilepsy in English, along with Christina Reshka, who you'll be meeting in a few minutes. We founded Epilepsy in English with the goal of making cutting-edge scientific research into epilepsy accessible to absolutely everybody by explaining it in clear, plain English language. So this afternoon's session is brought to you by Epilepsy in English in collaboration with FutureNeuro, who you'll hear more about, DCU PPI Ignite, our patient advocate Maria, Science Foundation Ireland, and of course, Epilepsy Ireland, who you all know a lot about. So a couple of months ago, just to give a bit of context for this afternoon's session, Epilepsy Ireland asked all of you to vote in an online survey about which research topics you most wanted to hear about and learn more about. We went away and added up all of your scores, and then we're delighted today to be able to offer you 4 talks from some of Ireland's leading experts in temporal lobe epilepsy, gene therapy, SUDEP, and epilepsy self-management. And then at the end of today's talks, we're very excited to be able to announce a new series of Epilepsy in English workshops, which are designed so that you can tell us more about what you hope research can offer to people with epilepsy. So please stay tuned until the end of this session for more details from FutureNeuro's Kira Courtney. So FutureNeuro, just to tell you, is the SFI Research Centre for Chronic and Rare Neurological Diseases. And one of our objectives within FutureNeuro is to change the patient journey through research which is informed by the needs of patients and neurologists alike. So we do hope you'll consider joining us for these workshops in November to help us to set the future research agenda together. An important note before we begin is I do have to say everything you see over the next nearly couple of hours is preclinical research. That means it can't be and must not be taken as clinical advice. We will endeavor to answer any and all questions about our research that you ask, but unfortunately, our speakers are not in a position to respond to any clinical queries. And if you have anything like that arising from the session, we would encourage you to talk with your neurologist. So with that all said, I will stop talking. I'm delighted to hand over to Christina Reshka, who will be chairing today's session and introducing our speakers. I hope you enjoy the session, and I very much hope to see you at these workshops in November to set the future epilepsy research agenda together. Christina.
Thank you very much, Gareth, for the kind introduction. Good afternoon, everyone. It's a pleasure to have you all here. And it's a pleasure to have these great speakers to bring the information to you in a plain English. So as Gareth already said, I echo all my words. And without further ado, I'm going to introduce our first speaker in the first theme. That's Professor David Henschel. That's going to speak about temporal lobe epilepsy. So Professor David Henschel is the director of FutureNeuro in the Science Foundation Ireland Research Centre. As professor of Department of Physiology and Medical Physics at Royal College of Surgeons, and his research focused on epilepsy, in particularly understanding causes and developing new treatments and diagnoses. In his spare time, to join a personal touch, David enjoys running, coaching soccer, and vegan cooking. David, the word is with you. Thank you very much.
Thank you, Christina. Can you hear me okay?
Yes, yes.
Great. So good afternoon, everybody. Thank you for the kind introduction. It's a real pleasure to give you a short overview of some of my own personal research interests, which concern the form of epilepsy called temporal lobe epilepsy. And I look forward to giving you a brief overview of those interests. And then later on, we will have a workshop which will go into much more detail. So the purpose of today really for me is just to give you a little flavor, a taster of the type of interests that I have. So just briefly, a little bit about myself. I'm a basic scientist. I trained in a discipline called pharmacology, which is all about understanding how drugs work, what drugs do to the body, what the body does to drugs. And there is, of course, an enormous need for better treatments for all diseases, but obviously my interest is around epilepsy. I grew up in the UK. I studied at Bristol University, moving to then Edinburgh for a PhD. I then moved to the US for about 6 years, and I came to Dublin in 2004. So I've been at RCSI now for over 17 years. And it was during my time in the US that I began to become interested in epilepsy, initially interested in the effects that seizures have on the brain, and in some cases trying to think of ways we might protect against those effects. But I'll talk a little bit more about more recent research shortly. So my laboratory uses a number of different techniques to study epilepsy. So we have models of epilepsy, we have cell culture models, so we can grow brain cells in dishes, and we also have access to samples which are donated by patients with epilepsy. So that might be a blood sample, for example, and in some cases there may be brain tissue samples. I might mention a bit more about that later on. We can then use powerful techniques such as gene sequencing technology to try and discover new models, new molecules that are present within the brain and that may be involved somehow in the disease process. And we can also make recordings of brain function, and this also helps us to understand what's going on in epilepsy. This is just a very brief overview of the lab. We have a fantastic team of scientists at RCSI. Some of these are PhD students working on understanding new mechanisms of epilepsy. Some of them are postdoctoral researchers. That means they already have their PhD, they're trained, and they're going into their final stages of advanced training. And we also have research support and laboratory management. And you can see on the diagram in the center, there's Gareth. Gareth has actually recently moved to set up his own laboratory, so I'm very proud of how well he's done. So my own interests focus on temporal lobe epilepsy. There are many different forms of epilepsy, and I'm sure those listening are aware of this. We see forms of epilepsy that emerge in childhood. Sometimes the cause is thought to be genetic. Other times, epilepsy development occurs in old age as a result of injuries to the brain, such as stroke or other conditions. But the most common form and the most difficult that we see to treat in adults is temporal lobe epilepsy. Seizures in temporal lobe epilepsy come from the temporal lobe, and as you can see on the picture on the left, this is this green area at the side of the brain. And the temporal lobe does a number of jobs for us. It's involved in the processing of sensory information in particular. It has roles in memory and also speech. One of the most important structures within this region of the brain is called the hippocampus. And you can see on the right-hand side, these two green areas highlighted. This is the hippocampus. This is a specialized region within the temporal lobe that's particularly important in memory and also spatial awareness. So as we move around, this part of the brain is firing away, telling us where we are in space. When a seizure occurs in somebody with temporal lobe epilepsy, it activates and also disrupts the function of this part of the brain. And this is why patients with temporal lobe epilepsy sometimes experience what we call auras or senses of deja vu. But this is actually occurring because the electrical activity within the brain is eliciting some of the normal functions of the temporal lobe. So you can experience an unusual smell or taste or a strange memory. Sometimes when patients have seizures from their temporal lobe, they may lose consciousness, but they may also remain conscious. So that's a little bit about temporal lobe epilepsy. But why do seizures happen in the first place? In the top left, you see a little cartoon of two brain cells, two neurons talking to one another. So our brain is made up of hundreds of millions of these cells connected to one another. But there are two basic types of brain cells. Some cells cause other cells to fire. We call these excitatory neurons. Other neurons actually dampen down activity. We call these inhibitory neurons. And for the brain to function normally, there must be a delicate balance between the excitatory neurons and the inhibitory neurons. And what we think happens in epilepsy is this balance becomes altered. We either have too much excitation or we have too little inhibition. And as a result of that, groups of neurons can fire together. They can fire in an excessive way, a little bit like an electrical storm. And I'm going to show you a short video now, which is a very slowed down version of what happens in the brain during a seizure. What you're seeing is recordings of groups of neurons, and you'll see a sudden onset of very high frequency firing of these neurons, almost like an electrical storm, and it spreads through this neuronal network. And after a short period of time, this activity dissipates. Most seizures last no more than a minute or two. There are natural mechanisms within the brain to switch off a seizure. So how do we treat it? Well, the main way that we treat seizures is to more or less dampen down the activity of the brain. We can do this by taking what are called anti-seizure medicines. These seizure medicines interfere with the signals that occur between brain cells, and they reduce the ability of these cells to fire altogether. But they don't work in everyone. In some cases, brain surgery might be an option for people who have very difficult to control seizures, and actually temporal lobe epilepsy can be effectively treated by a surgical operation. But this type of surgery is not available for everybody. One of the other things we know about current treatment of temporal lobe epilepsy, and indeed many other forms of epilepsy, is that despite all of the new treatments that we've developed, and the field of epilepsy has been very successful in this regard, we have more than 20 different medicines that can stop seizures. But despite all of the newly developed medicines, we still find about 1 in 3 patients for whom seizures continue. And this hasn't really changed in the last 50 or more years. So we need to think differently about how we're going to treat epilepsy. We need to reduce the numbers of drug-resistant patients so that more patients have full seizure control. We need to start to develop drugs that actually modify the disease. All of the seizure treatments that we have so far simply dampen down brain excitability. If you stop taking the medicine, then seizures will return. We also don't have anything we can give somebody who's at risk of developing epilepsy. Perhaps somebody who's had an injury to the brain, so that we can give them a treatment after that injury that will prevent them ever developing epilepsy. And these are the type of questions that my lab is interested in. So we've been trying to think differently about how we might treat epilepsy. And for temporal lobe epilepsy, one of the big challenges is that there are multiple changes within the brain occurring. There's not a single gene that's at fault here. Several different processes appear to be happening within the brain at the same time. We see changes in cell number, we see inflammation, we see changes in the connections between cells and even the structures of the cells themselves. And I think what we have come to the conclusion is that targeting one of these processes on its own is never going to be enough. We actually need to approach this problem by thinking of additional ways to change gene activity. If you take, for example, a set of genes which are known to control one of these processes, there are dozens of genes active at the same time. So just going after one of these genes might never be enough to stop temporal lobe epilepsy. So we have a multi-target problem. So could we find a multi-target solution? This comes on to one of the key areas of research in my laboratory. And the work that we do actually traces all the way back to a discovery made in 1993. This was about the time when I was entering my final year of undergraduate study. a group in America was studying the development of a small worm. The worm is called C. elegans. It's a type of nematode worm, and it's a fantastic model organism for understanding development. In fact, we humans share many, many genes in common with this simple worm. So by studying the worm, you may learn new things about humans. What they discovered was that there was a new type of genetic material that was produced that had never been noticed before. And this type of genetic material was sort of sticky. It used to stick on to other genes, and by doing that, it would affect how they worked. The discoveries that were made in 1993 were then ignored for nearly a decade, until they were rediscovered and found to be present also in humans. And this triggered an enormous amount of research, which led on eventually to a discovery that these same molecules or similar molecules that were found in this worm are actually active and potentially involved in epilepsy. These molecules are called microRNA. They have a very distinct structure. You can see this sort of double strand with a little loop at the end. And as genes are being activated within the nucleus of a cell, these molecules come in and they regulate how that process occurs. And now back to that multi-targeting question, we know that a single one of these molecules can stick onto 100 or more other genes. So if we can find that one molecule that's important, we might be able to regulate 100 other genes at the same time. So this gives us an enormous power to change gene activity within the brain. I'm going to show you a little graph which actually shows this in principle. We've been able to design little synthetic molecules that can bind onto these fragments and stop them from working. And in a model of epilepsy, we've been able to show that we can reduce seizure-like activity for long periods of time following a single treatment with this molecule. So what we're trying to do now is understand if these new types of drugs can work in human models. Can we prevent seizure activity in human models? We're also trying to find out how they work. Obviously, because an individual one of these molecules can regulate levels of 100 genes, it's important to try and understand how that happens, and it might help us try and make them even safer. And finally, we're thinking about new ways that we can deliver these treatments to make them work longer, more safely, and we're exploring gene therapy approaches. So thank you very much for your attention. I hope that has given you a bit of interest and insight into the work that we're doing in my laboratory, and I'm happy to take any questions.
Thank you very much, David. It was a fantastic presentation. I really would like to encourage everyone to add their questions to the Q&A. And as Gareth mentioned before, if we're not able to answer all your questions today, we're going to bring them anonymously, and we're going to bring to the workshops for further discussion. And as David just said, this today is just a taster of what we're going to touch in the workshops. So I'm going to do the matter of time, David. I'm going to start, and I'm going to ask you as the director of FutureNeuro, and with the prominent position that you have in the epilepsy field, how do you see epilepsy research for the future, and how can we reach what we really want, that is closer and closer to translation?
Well, that's a very big question. I mean, epilepsy research spans such a wide variety of work from, and I think the FutureNeuro Center tries its best to capture the spectrum of research. So everything from the level of the gene all the way through to the patient experience. And I think what I would say is I've never seen a more exciting time than the last few years and into the future in terms of how technology is transforming how we can study the brain. In the past, the tools that we had were very kind of crude and simplistic ways of recording activity. We're now able to record individual brain cell activity. We can measure millions of molecules within a single brain cell at the same time. The technology we have and the therapeutic approaches we can use now are phenomenal, and it's just a very, very exciting time. I think in the near future, we're going to be able to do things that were impossible in the past.
That's brilliant. We have a few extra questions. So I have a first question actually about gene therapy, but if you allow me, I'm going to skip that one, and we will answer soon after. Discuss with keep it for later. So what's the percentage of temporal lobe epilepsy is successful, and what are the potential side effects? I would say surgery, possibly.
Surgery? A question about surgery?
Ian, if you could just type, would be surgery? If
it's a surgery question, yeah, the success can be very high. So it all depends a little bit on whether they can identify a particular part in the brain to remove. Some patients with temporal lobe epilepsy have a clear sort of area in the brain. If that's removed, then it can cure epilepsy. In fact, not just reduce seizures, it can actually cure epilepsy in some cases. And certainly, I think about 80% of patients with that type of surgery have dramatically improved seizure outcomes. The problem tends to be when there's nothing obvious on a brain scan. It makes it a little bit harder to know which part of the brain network to act on.
Thank you, David. There is another question from Michelle here. Are these areas in the brain associated with temporal lobe epilepsy same as those that are affected in autism? So what would be the relationship?
So I would have to say, first of all, I don't know all that much about autism, but my understanding is that the problems in autism tend to occur more towards the frontal part of the brain rather than the temporal lobe. But autism is still not well understood in terms of the kind of the cellular changes and the molecular changes. And it may be that there are small changes across other brain regions. So unfortunately, I can't give a very good answer to that.
That's brilliant. Thank you, David. We have some extra questions, but as I mentioned, we're going to keep for the workshops. We will get all them answered later on. Okay, we're going to move now in a matter of time to our next speaker. So I would like to introduce Professor Matthew Campbell, who is a FutureNeuro Principal Investigator and Associate Professor in the School of Genetics and Microbiology at Trinity College, Dublin. So his laboratory focuses on trying to understand the role of brain's blood vessels in health and disease. And the lab uses the power of molecular genetics to unravel how the disruption in the integrity of the capillary network in the brain is a key driving force in epilepsy. By better understanding how the disruption of these blood vessels can drive seizures, we may be able to develop new forms of therapies for epilepsy. In his spare time, Matt likes to spend time with his children, Callum and Olivia, and also play tennis and cycling. Matt, thank you very much for joining us today, and the word is yours.
Great, thanks, Christina. Can you hear me okay?
Yes.
Perfect. And you can see my slides okay, I presume, as well?
Yes.
Great. So, look, thank you very much for giving me the opportunity to speak today. It's always a real privilege to speak at these patient forums. I'm by no means an expert in epilepsy, but since I joined the FutureNeuro Center as a principal investigator a number of years ago, I've really started to learn a lot about epilepsy, and I've taken a lot of the learnings from my apply them to epilepsy. One of the areas that I have a lot of expertise in is the concept of gene therapy. I'll talk about that a little bit later on, but what I want to do initially is give you a flavor of the work that I do in my lab, how it applies to epilepsy, and how we've used the patient experience and patient participation in research to really formulate ideas as to how we might be able to treat, especially temporal lobe epilepsy, moving forward. So taking a look at these standard medical textbook images that you see on the left-hand side, you can see that the brain has a lot of blood vessels. So the reason the brain has a huge amount of blood vessels is that, unlike smaller species and smaller animals, the human brain consumes about 1/4 of our daily energy intake. So when you log off this later on, this forum, and you're sitting having your lunch or having your dinner later on, 1/4 of the plate, the energy within the food that's on your plate, is being used simply to make your brain work. And that's just working at baseline. Because the brain has so many neurons, so brain cells, we've got about 100 billion brain cells in a human brain. We estimate that because the energy demand is so high, the brain cannot store glucose. It needs to get a constant supply of glucose. We estimate that essentially every single one of those 100 billion brain cells has its own capillary. So capillary is just a small blood vessel, a microvessel. So no neuron, no brain cell is ever more than about 20 microns away from the capillary. And this basically means that every single brain cell has its own capillaries. It's perfused, it's served by its own brain capillary. So when you think about that in the context of neurodegeneration, where a neuron is dying, or in the context of epilepsy, where a neuron is misfiring, it's producing too much electricity or sometimes too little electricity, we have to also think that because every brain cell has its own blood vessel, what if the disruption was coming from the area of the blood vessels as opposed to solely coming from the neurons? What if the disrupted blood vessel, maybe the blood vessel might be leaky? Could that be propagating and driving the seizure? And if we can prove that that's happening, maybe are there ways to tighten up the blood vessels and stop them from being leaky that could actually prevent seizure activity? A few other facts of the blood vessels in the brain. The capillary network is so vast that if you were to take out all the capillaries and stretch them out side by side, they would stretch from Dublin to Paris. If you were to look at the blood vessels in the context of the surface area, they'd have the surface area of a tennis court. So we're talking about a really, really huge capillary network in the human brain. And these type of medical textbook images don't really do justice to the sheer number of blood vessels that are in the brain. What we need to do is look at things like a vascular cast. So this is a vascular cast of a human brain that was prepared by my colleague Peter Guyard in the Netherlands. And what you're looking at here essentially is the blood vessels within the brain. So in a cadaver, a polymer has been injected into the vascular network of the cadaver, and the brain has been removed from the brain material, has been digested, and what you're left with is just the blood vessels. And it really gives you a strong impression of the sheer density of blood vessels in the human brain. And to think that if any of these blood vessels become dysfunctional, that they could drive disease, be it neurodegenerative disease or conditions like epilepsy. So taking this background, my lab really tries to focus on understanding these blood vessels in health, but also disease states. So we work a lot with animals, but we also do a lot of clinical research. And what I wanted to do today is show you some images from a recent project that was initiated by Colin Doherty. So everyone here knows Colin. We had the concept that a lot of patients who are being worked up for neurosurgical resection, so these will be patients who have treatment-resistant temporal lobe epilepsy, who are being worked up for surgery either in Beaumont or down in Cork. And what we've done is we've asked patients to get involved in research whereby we will look at the blood vessels in their brain before and after surgery to see whether or not these blood vessels are leaky or not. Because myself and Colin had this hypothesis, this idea that maybe one of the key driving forces of treatment-resistant temporal lobe epilepsy was that the so-called blood-brain barrier, so the properties of cells that line blood vessels in the brain that protect the brain from toxins, maybe that was disrupted and that was propagating and driving the seizures, and that's maybe why some patients were becoming resistant to treatment. So what we did was we invited about 8 patients before their surgery to come into Colin's clinic and have a brain scan, so have an MRI scan. While the patients are lying in the scanner, they're injected with a dye that lights up the blood vessels. Now, in the normal, healthy human brain, this dye will just simply move all around the blood vessels in the brain, and then it'll wash out essentially. So you'll get a very, very low signal. But in the context of a leaky blood-brain barrier, where the blood vessels are leaky in any way, this dye will leak out of the blood vessels and deposit in the brain and allow us to see it. It'll give a little signal that allows us to see whether it's in the brain or not. So let me show you some of the images that we've collected from this study. And just to reinforce that this is the type of study that is impossible without patient participation. And it's really patient-driven. You know, patients want to get involved. They want to understand how their condition is propagating and developing. So this type of work is actually impossible without patient participation. But also, as you'll see in a minute, the ideas that we have formulated for gene therapy in treating epilepsy have actually come from this patient participation. So what you're looking at here is a young man who has been having more than one seizure per week for nearly a decade of his life. Okay, so very, very debilitating. When he's in the scanner, we inject the dye, and anywhere where you see red, you're seeing leaky blood vessels. So it's pretty clear that even though the seizures are emanating from the temporal lobe, this region where you can see me pointing out in with the cursor, there's leakage of blood vessels throughout. And we think that this is a key driving force in the pathology, and maybe a key driving force in why this individual is having a lot of seizures. Now, look what happens after the temporal lobe is resected. So this young man went and got surgical resection in Beaumont Hospital, and we got him back into the scanner within about 4 or 5 months after the surgery. And this is what we see. So we see an almost complete resolution of the leaky blood-brain barrier throughout the brain. So while the individual is completely seizure-free post-surgery, it's worked very, very well. It's a classic example of how these surgeries work so well. What's really intriguing from a scientific point of view is that hand in hand with this seizure-free status post-surgery, the blood vessels are not leaky anymore. So the concept now is, well, could we potentially target therapies that could regulate the integrity of the blood vessels to prevent seizures? So rather than having to work towards and move towards surgery, could we actually find a way to treat an individual like this by stabilizing the blood vessels? Because it's very, very clear that the surgical resection is stabilizing the blood vessels throughout the brain. So maybe could we contemplate ways to stabilize the blood vessels without the need for surgery and thereby treat epilepsy? So very, very novel concepts and really driven by the patient participation in this research. So what does all this have to do with gene therapy? Well, let me talk briefly about gene therapy. And this is really just a taster prior to the workshops in a couple of months that I wanted to talk about what gene therapy is. So gene therapy is a very fast evolving form of therapy. In general, gene therapy can traditionally be thought about as a condition where we can simply replace dysfunctional genes. So in very, very rare forms of epilepsy or rare forms of eye disease, which we work with a lot, where we know the causative gene, we know that the gene that is causing the condition is dysfunctional. What we can do is we can take a protein shell, which we term an adeno-associated virus. So people know about viruses now, and all it is essentially a protein shell that contains genetic material. The virus of choice that we use it's like an attenuated virus. It's called adeno-associated virus. It's very, very small, 22 nanometers. And we can engineer these viruses. So we can hijack the genome, the DNA that's inside this virus. So unlike the SARS-CoV-2 that causes COVID-19, this is a DNA containing virus. And it's been tailored such that it infects neural tissue. So it infects cells that don't divide, like neurons or endothelial cells that line blood vessels. And the idea has traditionally been to package a functioning gene in the context of rare disease, and then inject the gene either directly into the brain in patients or into the eye in the context of blindness, or to inject it systemically, so to inject it intravenously. Now, what our concept with gene therapy in our lab is that we have identified components of blood vessels that cause blood vessels to tighten up or to be very, very leaky. And what we're actively doing, and we'll talk about this in the workshops, is we're looking at ways to introduce the genes that produce the protein that are essentially like glue for blood vessels into an adeno-associated virus. The idea then will be that we will be able to stereotaxically, so in very, very site-specific regions of the brain in patients, we will hopefully be able to inject these adeno-associated viruses to restore the integrity of blood vessels. And by doing that, we hope that we'll be able to stabilize the blood-brain barrier and thereby treat epilepsy. So working towards something like a surgical resection, but without the need for surgery. So this is just a sort of a brief synopsis of what we're doing in my lab in the context of gene therapy. And I'm very happy to stop here and take questions on other approaches to gene therapy, if there are any questions. So thanks, Christina.
Thank you very much, Matt. It was a brilliant talk. So I would say that we have the question that actually has been asked a little bit earlier as regards gene therapy. I know that Professor Henschel already mentioned something in the chat, but I would like just to bring up here again. So we had a question from Rupert. When do you think that gene therapy treatments might be available, if ever? So what's your vision on that? Okay,
so I'll talk first of all just about non-epilepsy related conditions. So gene therapies are available for some conditions. So Novartis, the company Novartis, have a gene therapy for spinal muscular atrophy, but also for a very rare form of blindness called Leber congenital amaurosis. These were approved by the FDA in the past 4 years. So this is happening, and these are real. So gene therapies used to be the remit of, all developmental type of approaches to therapies, but there are now gene therapies being approved by the FDA in America. In the context of epilepsy, it's still relatively early days, but because there's a paradigm has now, a precedent has now been set that the FDA know how to deal with the development of gene therapies, that things should move quicker for other conditions like epilepsy. So we always throw out the phrase of 5 or 10 years. I can't put a figure on it because it's going to be context dependent. So for rare forms of epilepsy, the chances are that there may be gene therapies quicker for the more common forms of epilepsy.
Yeah.
That tends to happen with genetics, that the rare forms of disease, we know the genetic cause, but the common forms of disease, we don't know the genetic cause because it's probably multifactorial. But suffice to say that there will be gene therapies for epilepsy in the future. I don't know when, but I've no doubt in my mind. There's just too many companies involved in it for us not to see a gene therapy for epilepsy in the future.
That's brilliant. Thank you, Matt. The next question actually is a way broader question. So from an anonymous attendee, what ever happened to the Human Brain Project and Obama's Brain Project in America? Does FutureNeuro work in a big project like this? If there is any money put into the understanding of the brain? I know, Matt, that's going to be very tricky for you. I would say we can even leave for the whole panelists and to discuss later on. I'm not sure you would like to comment on that. Otherwise, we can just move and we leave for a broader discussion later on.
That might be, yeah, we might leave that one for a broader discussion. Yeah,
that's fine. How did surgery resolve the leaky vessels in other areas of the brain?
So, so that's a really, really good question. But my simple answer is I don't know. But I've got some what I think are good ideas. So what I think happens in the brain where you have a catastrophic breakdown, a focal point in the brain where the blood-brain barrier becomes really dysfunctional, what I think happens is that it propagates, it spreads. So it's very similar to a traumatic brain injury. If you get a focal point of a traumatic brain injury, say a penetrating injury to the skull, generally what will happen is you'll get brain swelling throughout the brain because you've got catastrophic blood vessel leakage in one focal point in the brain, but it spreads. And I think that's what's happening in epilepsy as well, that the seizures are likely driving the blood-brain barrier disruption, and that disruption becomes so catastrophic that it spreads throughout the rest of the brain. And that's the working hypothesis.
That's brilliant. I would say there is another question here that would go that would marry together with that from Michelle. So she's asking here: is there much research on the links between ADHD, ASD, and epilepsy, and how can you talk a bit about susceptibility of other conditions? If you'd like to talk about your research in a kind of broader way as regards other conditions.
Yeah, so I mean, humans are funny creatures in that we like to label things, and so it's the same with all disease. Diseases are they're termed diseases because they're given names because we as humans give them names. So absolutely, there's likely going to be a spectrum of conditions. So there's quite a lot of crossover, for instance, between epilepsy and psychosis, and so are they just different? Are seizures or visual or auditory hallucinations, are they just different manifestations of the same biological process that's affecting different parts of the brain? So absolutely. And obviously from my perspective, my lab is really focused on blood vessels, and we always try to figure out how the blood vessels become dysfunctional in brain diseases. We've got a lot of data to suggest that, especially in neuropsychiatric conditions like bipolar disorder, major depression, and schizophrenia, that there is a dysfunction in blood vessels quite similar to epilepsy, not as catastrophic as epilepsy, but very, very similar. So I think your thought process there is really, really on track with the way that we're thinking, that there's huge overlap between these conditions. And if we can understand that overlap at a molecular level, again, that can throw out new therapeutic targets and potentially lead to new drugs.
That's brilliant. Thank you so much. We have more questions, but we're going to see if we can have time at the end of the discussions. Otherwise, we're going to bring for the workshops. And thank you so much, Matt, again, for joining us today. So we're going to move to our next speaker today. That is Dr. Yvonne Langen. She's going to talk about her research on SUDEP. She's a consultant in clinical neurophysiology in St. James Hospital and senior lecturer in Trinity College Dublin. She was appointed as a consultant in clinical neurophysiology in St. James in 2009. Dr. Langen has a special interest in epilepsy and spent 3 years in the Department of Clinical Experimental Epileptology in the Institute of Neurology in London, where she was involved in research into mortality in epilepsy, which remains a significant research interest. In her spare time, Yvonne sings in her local choir. Thank you so much, Yvonne, for joining us, and the word is with you.
Thanks, Christina. Thanks for that lovely introduction. I'll just go back to the start of the slides there. My name is Yvonne Langan, and I'm a clinical neurophysiologist in St. James's Hospital, very much involved in the clinical end of things. And I'm going to talk to you today about sudden unexpected death in epilepsy, SUDEP, and really try and share with you my sort of SUDEP journey and emphasize the contribution that people with epilepsy in Ireland have made to the body of knowledge. It's a difficult and sensitive subject, but we will have a workshop on the 9th of November. And, you know, even though it is a difficult subject, I would encourage you to register for the workshop. And Kira will supply the registration details at the end of the session. So I first encountered a case of SUDEP way back in the midst of time when I was a neurology registrar in St. Vincent's Hospital in the mid-90s. And, you know, it's such a shocking occurrence with an individual in the, you know, often in the prime of life taken from us, that it sparked an interest. And we went on then to look at the deaths that were occurring in our St. Vincent's population with epilepsy, covering the South Dublin and Wicklow area. And from there, it really led to an ongoing interest in SUDEP. And, you know, the sort of research into SUDEP really took off from the mid-90s onwards, I suppose. But, you know, if we look back in time, it was something that was recognized, you know, hundreds of years ago. I'm having trouble with the slides there. I'll just come out. Oh, there we go. So this is George Washington, and he had a stepdaughter called Patsy Custis, who had epilepsy. And as a teenager, Patsy died, and it seems to have been a case of SUDEP. We have the description that George Washington gave of her death. And he tells us that she rose from dinner about 4 o'clock in better health and spirits than she had been for some time. And then she was suddenly taken with one of her usual fits, and she expired in less than 5 minutes without uttering scarce a groan, a word, or a sigh. And my poor dear wife has been completely laid low by this sudden and unexpected occurrence. And I mean, this is a story that resonates with us even to this day. This is something that happens to patients with epilepsy. And what is it? How do we define SUDEP? Well, we have a kind of a definition that says that sudden and unexpected. They can be witnessed or unwitnessed deaths, not due to trauma or drowning, and where the post-mortem does not demonstrate a cause for death. So basically, sudden death in people with epilepsy with no cause identified. Well, how common is it? Well, overall in the population with epilepsy, it's occurring about 1 in 1,000 per annum. But this does vary according to the groups that are examined. And our own study from St. Vincent's Hospital is in this graph here, it's number 11, where we determined an incidence of 1 in 680 per annum. But the incidence does vary according to the group looked at. So in large population-based studies that will have both patients with well-controlled and more difficult epilepsy, it's coming in at roughly 1 in 1,000 per annum. But as we look at groups with more difficult to control epilepsy, we can see that the incidence rate rises, maybe as high as 1 in 100 in patients on epilepsy surgery programs. So we're getting hints from these studies that, you know, might lead us to identify potential risk factors for SUDEP. And this work then led on to me being able to go to the Institute of Neurology in London and take part in the UK case control study of SUDEP. And So case control studies were the next obvious step in trying to identify risk factors, where we would compare individuals who had died through SUDEP with individuals with epilepsy who were still living and try and pinpoint differences. So there've been lots of case control studies. And this is a work that combined the results of a number of them, including our UK case control study. And I suppose the most important or the most standout risk factor for SUDEP is poorly controlled generalized tonic-clonic seizures. Well, what about the mechanism? Well, we don't actually know what the mechanism of SUDEP is. And indeed, it's possible that it's not just down to one single mechanism. I suppose the most obvious thing that people might think of are cardiac abnormalities. And we do know that after a seizure, there can be abnormalities of heart rhythm. And in very rare cases, there can be stalling of the heart. So I suppose this was one of the first things that people thought about when it came to the potential mechanism for SUDEP. And indeed, we were lucky enough, myself and Aoife Laffin, who's there in the photograph receiving a prize for the work, we were lucky enough to have the support of Epilepsy Ireland and the Barbara Doyle Memorial Fund to enable us to look at baroreceptor sensitivity in epilepsy. And baroreceptors are involved in the autonomic nervous system, which might control the heart, if you like, and heart rhythms, and abnormalities of that process might predispose an individual to abnormalities of heart rhythm and stalling of the heart. And we did find increased baroreceptor sensitivity in individuals with epilepsy. And that's sort of adding to that whole body of knowledge on potential cardiac abnormalities. What's interesting, though, is that in any deaths that have been captured in epilepsy monitoring units, for example, there often seems to be an abnormality of respiratory function with oxygen levels dropping in the blood, and then this might be followed by an abnormality of the heart rhythm or a slowing of the heart. And that's certainly a big area of research interest for some labs. And there's some suggestion indeed that certain medications might help to prevent respiratory depression after seizures. But the largest study that called the Mortimer study that captured SUDEP deaths in epilepsy monitoring units actually seems to suggest that maybe it's a centrally mediated process where abnormality in the brain causes the, I suppose, respiratory center to act up a bit, respiratory depression, and then subsequent abnormalities of heart rhythm. But of course, there's still an awful lot of work to do and a lot of unknowns in this area. Some suggestions from animal models that neurotransmitters and chemicals like serotonin or adenosine may be involved in either helping to prevent or in fact promote sudden death. So a whole load of unknowns in that particular area. But that doesn't mean that we can't use our knowledge of risk factors to help reduce the risk for an individual of SUDEP, even if we don't know the exact mechanism. So this is a study carried out by the HRB. It's the National Drug-Related Deaths Index. And every year they go into coroners' offices and they identify, by looking at coronial records, deaths that have occurred in individuals who have problems with drugs and alcohol. And they identified a number of epilepsy-related deaths in this group. And this led really to collaboration, and we thought, well, gosh, they have a great mechanism of going into coroners' offices and identifying deaths. Wouldn't it be great if we could expand this and use it as a mechanism of collecting information on deaths due to epilepsy? So with the support of Epilepsy Ireland and the Irish Institute of Clinical Neuroscience, we are now working with the Health Research Board and piggybacking, if you like, onto their methodology. And in the process of identifying epilepsy deaths referred to the coroner in 2019. And I think it is important that we have up-to-date information on epilepsy-related deaths and SUDEP in Ireland and get an up-to-date incidence rate. And it may be that information from these deaths will, we may get be able to identify patterns. So very much looking forward to being able to analyse the information that comes out from that data collection. And it's interesting to think how it would never have happened if the guys in the HRB running the NDRDI hadn't been alert to the fact that they were identifying epilepsy-related deaths in that cohort. So that's kind of taking us from, way back into the past up to what we're involved at in the present, and then on to the future. I'm on the steering committee of a multi-centre international case control study looking at risk factors for SUDEP. And you might say, well, you've done, there have been lots of case control studies to date. Well, I think it's important that we try and, go bigger and better. There are questions that were raised in those case control studies, including, the potential protective element of supervision at night that need to be pinned down further. So this is, if you like, a bigger and better version where we're trying to get a large number of cases, hence the multi-centre and international nature of the study, and to be very sort of, I suppose, exact in the questions that we ask to try and glean as much information as possible. We haven't got ethics committee approval yet to include Irish cases in this study, but I'm very much hoping that this will be coming soon, and we're also very much hoping to use the epilepsy EPR as part of this study. We also, you can see the last name on the slide there, Robert Scragg, but he's an epidemiologist that was involved in the studies of sudden infant death that identified that placing babies on their backs reduced the risk of SIDS. So it's, this is great from our perspective, having his advice in terms of our study design. So you can see from, the work I've told you about there that, it would not have been possible without the input and support of people in Ireland with epilepsy and their families. So we really need your input and your support going forward. And for that reason, I would very much, again, like to encourage you to register for the workshop. Thank you.
Thank you very much, Yvonne. It was a fantastic talk. Thank you so much for coming and joining us today. So I'm going to ask you, once we are talking about genes a lot lately, is SUDEP genetic? How do you see the role of gene expression and looking at this function of our genes and hopefully predicting it?
Well, there have been some studies looking at genetics in SUDEP, mainly concentrating on abnormalities in sodium and potassium channels, postulating that a condition such as a channelopathy, which might predispose one to both epilepsy and potential cardiac abnormalities, might be involved in the genesis of SUDEP. And I suppose we just don't have enough information yet to come to any sort of firm conclusions. Certainly that case control study that I mentioned to you at the end, we are asking if the patients with epilepsy have had any genetic studies or any cardiac studies. We're asking if there are any samples available for analysis from both cases and from their families. So I think the general recommendations going forward are that, we should be thinking of getting genetic information from any of our studies that we do.
That's brilliant. Thank you, Yvonne. So we have another question. What are the simple measures that people can take to minimize the risk of SUDEP?
I think the number one measure has to be to optimize seizure control by whatever mechanisms are available or most appropriate to any one individual, be it anti-epileptic drugs, thinking about surgery if it's appropriate, reducing seizure triggers. So I think number one and most robust across all the studies, reducing seizure frequency of convulsive seizures. In terms of partial seizures, there isn't really much in the way of evidence that suggests that they significantly increase the risk of SUDEP. There are some other factors from case control studies and from other studies. One is that maybe supervision at night is helpful. We know that most SUDEP cases are unwitnessed. So maybe there is something about having a witness seizure that is protective. Maybe if there's someone there to give some stimulation to make sure that someone is breathing and all the rest of it. So there are some hints from work that supervision at night might be helpful, but that is only really in one case control study and from some other, descriptive studies. So that again is something that needs to be pinned down, but obviously for, as individuals dealing with your neurologist, it may be something that comes up for discussion.
Thank you, Yvonne. So we have another question. Is there any evidence of good nutrition and organ function and the ability of the body to absorb the medicine in reducing SUDEP?
Well, obviously, if an anti-seizure medication is not absorbed, it may not work. So if there are any medical reasons that prevent anti-seizure medication from being absorbed, obviously that may be relevant. There have, in studies that people have looked at maybe blood levels of various anti-seizure medications, and we do know that maybe having low levels or suboptimal levels of seizure medications in the system may increase risk. And that's really just because it is increasing the risk of having a seizure as opposed to being an independent risk factor. From the point of view of general health, there's no evidence that any one particular condition may increase the risk of SUDEP.
Thank you, Yvonne. So we have a question and comment from Peter Murphy. So thank you, Yvonne, for shining the light on SUDEP in Ireland and internationally for over 20 years. This new study with the HRB will be vital in understanding the scale of SUDEP in Ireland. What do you think we can do to tackle SUDEP in the information? Oops, no. What do you think that we can do to tackle SUDEP with the information that the study will gather? Is there enough of a conversation about SUDEP in epilepsy community between doctors, patients, and do we need a SUDEP strategy in Ireland specifically?
Well, I think it's such a difficult subject. I think the general consensus is that it should be discussed with people with epilepsy, but that's maybe easy to say and a lot harder to do because it is so difficult. We do know from some surveys of UK neurologists that, while it may be in the guidelines, that, a much smaller proportion than we might have anticipated discuss it with their patients. And also because it is such a difficult subject, even if there is a discussion, it may be that not all the information is appropriately absorbed. So I think, events like this are very important in increasing knowledge generally about SUDEP and maybe making it easier then to have that one-to-one discussion about, for me, what my SUDEP risk might be. So I think it's like a two-armed approach really in terms of information, increasing information generally, and also encouraging that one-to-one discussion between the patient and their neurologist, and making it easier. And I think the role of the specialist nurse and the AMP is really vital in this and the building up of that relationship. But there's no way to getting around the fact that it is a very difficult it's a difficult subject. In terms of the HRB study, I think people might have said, well, sure, why don't you just assume that you're the same as the UK in terms of incidence rates and all the rest of it. But I think we're coming from different health systems. And I think it is important that we have up-to-date Irish figures so we can see if any particular, if we've changed over time, if we're different to other populations that we might assume we should be the same as. Are there any particular patterns emerging? There's a recent study, I think it's from a US group that found actually that different socioeconomic categories have different risks. So I think that, it's really looking forward to analysing the Irish figures and seeing what patterns emerge.
That's absolutely great. Thank you so much, Yvonne, for joining us today. And we are really excited to hear more about this very, very important topic during the workshops. Thank you again.
Thank you.
Just in a matter of time, last but not least, we're going to move to our last speaker, Jack Banks. Jack is a PhD researcher in Trinity School of Medicine and FutureNeuro eHealth department. Jack is driving a collaborative research project between FutureNeuro and the S3 Connected Health. Jack's research is examining the needs and wants of people with epilepsy, their carers and healthcare providers with regard to their care. He's primarily based in Dublin Mid-Leinster Epilepsy Service at James Hospital, and his research role, he works alongside with neurologists, specialist nurses, and dietitians seeking to innovate in epilepsy care. Jack is from Limerick, and in his spare time, he plays hurling for Castleknock GAA and likes to DJ, read, and write about music. Over to you, Jack. Thank you so much for joining us.
Thank you very much, Christina. I really appreciate that introduction. So good afternoon, everyone. And yeah, thanks a million for having me here today. So I'm going to give a little talk here about monitoring your own epilepsy and just give a quick overview of the area that I've been researching for the last few years in order to achieve my PhD, and give you some information about a workshop that I'm going to be hosting in November. So like I was saying there, I'm a PhD candidate. I handed in my thesis at the end of August and I'm waiting to be examined on it. I'm supervised by Professor Colin Doherty, who was speaking earlier on, and I'm based within the academic unit of neurology in Trinity, and I form part of the eHealth team in FutureNeuro as well. So the area that I've been researching for the last 3 years is called self-management, and there's lots of definitions for what this term is. But there's a couple of, a really nice example I came across early in my PhD is, I was reading the work of a lady called Sarah Regare. So Sarah is an engineer from Sweden, and she conducts research, and she also lives with Parkinson's disease. And when she was researching her own condition, or when she broke down her entire calendar year, she found that she spends 1 hour a year in neurological healthcare, and the other 8,765 hours in self-care. So 99.9% of the time, she was at home, in work, out and about, living with her condition, and she was responsible for her own healthcare and the manner in which she conducted herself in order to stay well. And when I'm asked what self-management is, the definition or this is how I interpret it, and what I think it means to me anyway. I think it's the activities a person carries out in their day-to-day lives to improve their overall quality of life and reduce the number of seizures that they experience. And specific for epilepsy, there's a number of activities that have been spoken about and are regularly discussed by clinicians with patients and appointments and things like that. So adhering to medication, of course, is seen as very important. Keeping a log or tracking when seizures occur. And then there's lifestyle factors as well, such as getting enough rest and sleep, and maintaining a healthy or an active lifestyle involving exercise and diet and things like that. In the last 20 to 25 years, a number of digital health or e-health tools have come into the market to help people live with chronic conditions when they're at home or at school or things like that. In the 99.9% of the time, they're away from healthcare settings. I've given a few examples here. So I suppose widespread use of YouTube, and I suppose how prevalent and important YouTube is to so many people for gaining information. So things like seizure first aid, there's a load of different videos available that will educate people about this. The availability of apps, I've got a screenshot here of the Epilepsy Ireland app, which has a number of functionalities which allow users to log their seizures or get graphs of what seizures are like over time. Some individuals like to use spreadsheets such as Excel to monitor themselves when their seizures occur. I've got a screenshot here. This is the Pisces patient portal, which was developed by my colleagues in the eHealth department in RCSI. So this was, the portal was in a research phase for a number of years, and it allowed individuals to view their electronic patient records and their own medical details. And a number of studies were conducted about the effectiveness of this and what users liked and didn't like. And this is going to be moving forward into a new phase now. There are also a number of commercially available seizure detection devices. This is the Embrace 2 watch, which contains accelerometers which detect when seizures can occur by measuring different physiological signals. And yeah, these are available to individuals commercially. You pay a subscription fee, and it alerts family members to when seizures occur. And yeah, they're quite popular.
And also there's the availability of different websites. And here's a screenshot of the Epilepsy Ireland website, which contains some excellent information about a whole host of different aspects of living with epilepsy. And then I suppose in a wider sense, not just dealing with epilepsy, but just in terms of lifestyle, we're seeing in the digital space so many different tools are available to individuals now, which allow them to monitor their own lifestyle and their health. So here is a Fitbit, which it's almost like every single day that you go outside, you see more and more people wearing them, and they're really, really common. And the availability of Apple and Samsung watches as well. They're a bit more expensive than Fitbits, but they're connected to your telephone device, and you're able to get a little bit more functionality, and you're able to do a few more things there. I suppose with the onset of COVID, we've seen an explosion in telemedicine and individuals receiving their care from clinicians without having to go into the hospital. So the use of things like email and telephone appointments and video appointments has increased exponentially almost since the onset of COVID. And the popularity of apps such as Headspace and Calm, I suppose across society, we are becoming more mindful people, and everyone seems to have acknowledged and is trying to incorporate a bit more meditation or a bit more mindfulness into their lives. And the availability of Headspace and Calm, they're recommended a lot by clinicians and people in the space as being very good for this kind of thing. Particularly in relation to epilepsy as well. MyFitnessPal allows you to track the macronutrient content of your food, and to monitor the number of calories you're eating a day and things like that. And I know from speaking to Dr. Laura Healy, who's the dietitian in the epilepsy service in James's, for individuals who are following the ketogenic diet, MyFitnessPal is a wonderful resource to allow you to see how you're getting on and seeing the nutrients content of your food. And online as well, there's a number of educational platforms which exist to allow people to, I suppose, like there's far more to living with chronic disease than just managing symptoms. And there's the psychological element to it, and educational programs exist which allow individuals to, I suppose, gain some of these psychological tools to help them live with the condition optimally and have the best quality of life possible. And yeah, so my PhD research, I investigated the opinions of people with epilepsy, their families and clinicians towards using digital tools and technology to help them manage their condition. So through a combination of surveys, semi-structured interviews with participants and focus groups, I was privileged to listen to the experiences of people with epilepsy and their families in particular. And I suppose when I collated all of my results together and I tried to draw conclusions from it, I found that most participants were open to the idea of using technology to self-manage their epilepsy, or they would like it to play some kind of role. And some of the challenges surrounding existing technology were identified, and participants together in the group, in the focus groups and in the workshops, they identified opportunities where they would like digital tools to help them going forward. So some of the challenges that were identified, I've just listed two here. So memory difficulties were commonly expressed, or they were seen as a challenge for individuals living with epilepsy. And a sentiment that was expressed was some patients who experienced memory difficulties, they were like, this technology is wonderful or whatever, and if it allows me to track my seizures, that's great. But a lot of the time, I don't remember when seizures occur. So as great as this technology is, it's not useful for me because I have to manually input it myself. And some patients as well were just of the belief that existing technology which exists, so all of the online resources that were available, the wearable devices, the apps, the things like that, they just didn't believe that they would change their situation for the better, and this would lead to them not wanting to engage with technology consistently, excuse me. And then I've given two examples of the ideas that were generated in focus groups as well. Some of the patients said that they would love that if data was generated from a wearable device or an app, it would be great if that automatically went into their medical record on the electronic patient record. And it also struck me as when I was speaking to individuals and their families, just how integral family members and parents and partners were to helping manage epilepsy. And at the moment, the needs of family members and carers don't seem to be being met, and they seem to be ignored almost. So it's important that going forward, tools need to be developed which cater to the needs of family members and carers of people living with epilepsy. And Kira will be updating you on where to register your interest for the event, for my workshop, but it's taking place on Tuesday, November 2nd. And I'm just going to have a discussion with anyone who would like to join about self-management activities, digital and non-digital tools to aid with self-management, the barriers and challenges to self-management, and making self-management a priority for epilepsy care. And I would love it if you could join. And yeah, that's me finished now.
Thank you so much, Jack, for this great talk. And let's move here to our questions. Actually, we have a question that was left behind that we can start answering now and discuss with the broader group. So Aileen asked, how do we know that I have epilepsy during this sleep time? And she's also interested to know about the cost of these devices to buy in order to monitor her her seizures at night, if so.
Yes, so in terms of the cost, generally what happens is you pay a subscription fee each month. It varies on the device. A number of different options are available to you. They can be anywhere from between €20 to €60 a month. So they are quite expensive. And before buying a device or before taking this, it's highly recommended that you have a discussion with an epilepsy specialist nurse or a neurologist who you receive your care from, just because they have information, and given the type of epilepsy you have, they would be able to send you in the right direction about which one would work best for you.
That's brilliant, Jack. And we talk a lot about artificial intelligence and a number of other technologies that are kind of every day more present in our days. So how do you see the future of the epilepsy management and the integration, considering all the technology that is available now and in the future?
Yeah, I think that automatically generating data from patients, or patients' data through wearable devices or information that they input into apps, I think that's so important that it's automatically inputted into the electronic patient record. I found that from my own research, it's a major barrier on the clinician side, that I suppose the lack of patient-generated data which feeds into the clinical record, because patients are the experts in their own condition, and if they're provided with a platform that will allow them to give as much information as possible to their doctor in order for their doctor or their nurse to make the best clinical decisions possible, that's the future in my opinion. And of course, that raises issues of consent and how happy patients are to, I suppose, share their data with their clinician. But I think that if we're as transparent as possible, and patients, with the patient's consent, all of the data that they consent to being sent to their doctor or nurse is given, then I think that's a safe and it's an effective way of, I suppose, enhancing the patient-clinician relationship and improving epilepsy care going forward.
Thank you. And we have another question. If you could comment maybe on that, how close we are to developing technology that can predict seizures before they happen. Interesting talk yesterday about the dogs predicting seizures. Is it possible for technology to mimic whatever is happening with the dogs? So,
so that's probably a bit beyond my realm, I would say, as I'm not a basic scientist, but I think that we're getting there. I think that in terms of making this transition towards a more technological form of epilepsy care, involvement is needed from private industry and from academic research centres and from clinical settings as well, which we're very fortunate to have here in FutureNeuro, because expertise is needed in each of those domains in order for us to, I suppose, incorporate technology in the most effective and convenient manner. I wouldn't be able to put a timescale on it, but soon, I would say.
That's okay. It was very unfair of me to put you in the spotlight.
A politician's answer, perhaps, but I think sooner than expected is what I would say.
I'm sure that we now can discuss with the whole group. I would like to invite actually everyone to turn on their cameras, and if we can have a final discussion with our speakers. I would like to go through the remaining questions, and then we can have a broader discussion. Thank you so much, Jack. It was really, really insightful, your talk, and please keep with us for the questions. So we already kind of mentioned, if someone would like to comment in Eileen's question as regards epilepsy in sleep, like what would be the best tool for identification of seizures?
I suppose I might, I'll tackle that one just from a purely clinical perspective. You know, oftentimes if there's a bed partner or someone in the house, they may, you know, hear things or have evidence of seizures occurring. Sometimes depending on the type of seizure that's happening, you might wake up feeling very tired, as if you've gone few rounds with Mike Tyson. Maybe there's evidence of a bitten tongue or whatever. But beyond that, and depending on the seizure type, it's possible that seizures could happen without, you know, knowing about them or having any evidence of them the morning after.
Thank you, Yvonne. So we have one that's directed for Professor Henschel here as well. So how long does it take for discoveries made by Professor Henschel's research to progress to the clinic? Will that time be shortened in the future, improving technology? Will it be improved by experience of COVID vaccines, and what we need to get the technology to market this medicine as soon as possible?
Very good question. It's an impossible one to answer with any degree of certainty. Typically in the past, a new drug being developed would be somewhere between 10 and 20 years. But I think with technology, and particularly some of the types of molecules that we're working on, it's possible to design and test these quite quickly now. Now, this hasn't happened in the epilepsy field, but there was a case of a rare disease published last year in the New England Journal of Medicine, which is sort of the top journal in the world for new medical discoveries, where in 1 year, they identified a patient with a rare degenerative disease. They identified the mutation. They designed a sort of synthetic molecule to treat that mutation, and they gave it to the patient within 12 months. So there are examples now where there can be extremely fast responses, which I think is, you know, is fantastic for the future. Whether it will ever get like that way for epilepsy, we'll have to see. And as Matt Campbell said earlier, common forms of epilepsy will be more challenging to develop these types of gene therapies too. But I think the more that these types of advanced treatment appear for other diseases, the closer it'll be and the faster it'll be for epilepsy.
Thank you, David. So we have a next question. What is the possibility of fully coming off drugs after years after surgery? I'm off 95% of surgery worked years ago. So if anyone would like just to make a quick comment as regards outcome after surgery. I think that would be a tricky one from our panel. I think we just
I think this is one of those examples where we are best not to
exactly. We can come back offline. So let me see. I'm just lost here. Apologies. From a SUDEP point of view, is it vital to discuss this at early stages? How many new moms leave hospital without being warned about SID? Is it necessary a discussion in the maternity setting, even though the stats are low and also in this area? So,
well, I suppose that's for me. My assumption is that sudden infant death is discussed with all new mums, and they're given advice on how to reduce the risk, and that would certainly have been my own experience, but I cannot answer whether that happens in every case. Certainly from the point of view of SUDEP discussion, the general consensus is that it should be discussed, but I think we all probably suspect that it isn't being discussed with everybody.
Thank you, Yvonne. I would say that we have some thank you about the questions that were answered. Let me see. I have a comment from Jan. I need to drop off, but I would love to thank you all for this brilliant session. If you can email any requests to participate in the future studies, I'm very happy to volunteer. Very happy to hear that. I would say I just would like to close now the session with the panelists, because there is this question that actually just put Matt on the spotlight about the Human Brain Project and the funding in epilepsy research in a general way. Maybe I would say it would be easier to ask David if he would like to comment in a very broad and simplistic way where we are at with these big projects that we've seen on television and the news, and how we can link with that.
Yeah, so absolutely, there's majority of the epilepsy researchers that work on genetics and the brain are involved in very large international consortium. There's a phenomenal one called Enigma, which is led out of the University of California. So the team at Beaumont and RCSI, and probably other sites like Trinity and St. James, they all contribute patients who provide access to their brain scans. That data goes into there, and they've made incredible discoveries about what happens to the brain over time in conditions such as epilepsy. So yeah, absolutely, researchers in Ireland are involved in these big projects. The Human Brain Project, I'm not aware of a major role of any of the teams, but that may be different for the guys in the clinic. There's also a lot of involvement in major European projects, genetics in particular. There's international consortium which are now collecting hundreds of thousands of genomes from patients, and this is how we make big discoveries into the future.
Thank you so much, David. I would like to thank you all for joining us today, and it was absolutely brilliant to have just this taster of what is coming in the workshops. I'm going to give the word to Kira, who is going to introduce the workshops and give more details. Just as a little comment here, Aileen and the other questions that we could not answer, we're going to be in touch with people, and the answers are going to be we're going to follow up in the coming days, and more of those are going to be answered during the workshops. So thank you very much for joining, and I'm going to give the word to Kira right now to give details about the workshops.
Hello everyone. So first of all, just thank you so much for your time today, and to the panel, of course, but most importantly, all yourselves at home for joining us. Without you, we can't do our work, and that's the most important message that we'd love you to take away today. But most importantly, dates for your diary. So we are running 4 workshops in November. I'm just going to make this a wee bit bigger. In November, starting on the 2nd of November with Jack monitoring your epilepsy, the 9th of November, SUDEP with Yvonne and Colin, 17th of November, temporal lobe epilepsy, David Henschel, and the 26th of November, Matt and Christina will be taking care of that workshop. All our workshops are taking place between 6:30 and 8 o'clock in the evening. You can register your interest of which workshop that you would like to attend to our website. It is www.EIE, so Epilepsy in English, it's short for Epilepsy in English, workshops.ie. Please bear in mind that places are limited, so I definitely suggest getting on there and selecting which workshop you would like to attend. And most importantly, that website is now live, so you can select which workshop you would like to go to. But the one thing I would suggest is, let's say you wanted to go to all 4 and you're interested in all 4 topics, you're more than welcome to join us, but definitely select the one that you really want to the most, should I say. So select that one first. Just a huge thank you to everybody today particularly everyone in Epilepsy Ireland who hosted us today after a number of days at your national conference. And I know it was a huge success. And like I said, everyone, all our panelists here today, thank you. I know it's Saturday afternoon and I hope everybody has a lovely day. And we are really excited to meet you all in November and to be part of setting the future of our research agenda. Thank you very much.